We know from twin studies that the vast majority of ADHD is due to genetic differences. On average, 80% of the differences in people in this room in their ADHD symptoms are due to differences in their genes. And in the last few studies, it was as high as human height, about 91%. Twin studies allow us to calculate how much of a trait is due to the rearing environment, and it's 0. All 40 twin studies published in the last 20 years have shown that the rearing environment has no influence on this trait. Yet the public believes that ADHD originates in bad parenting. The twin studies also tell us that there's a small percentage of ADHD that's arising from non-genetic causes, and those are those acquired injuries that we talked about earlier. And just in case you were wondering, yes, we are discovering genes for ADHD. We have had more success in this area of psychiatry than with any other disorder that I know of. We now know from genome scans published just last year, where we've scanned all 35,000 active human genes for ADHD genes, that it's going to be about 20 to 25 sites. And out of those 20 to 25 sites, we've nailed down about 5 to 7 of them. And here's a few. I want you to notice that there's a D in front of the first 4 genes. It means that those genes regulate dopamine in the brain. So no surprise, the genes that regulate dopamine are ADHD risk genes, and no surprise that drugs that improve dopamine in the brain work for ADHD, because those drugs are altering the effects of these genes in the brain, which means that psychopharmacology is a form of genetic treatment. Isn't that fascinating? It's not a Band-Aid, it's not a cover-up, it's not missing the point, it's actually helping to manage the underlying genetic problems, if you will, the genetic differences in the brain. Now, let me take one of these genes and explain it to you because it's kind of cool. I won't go through all the genes, but bear with me. This is a nerve cell that I pulled out of your frontal lobe. Ouch, I'll bet that hurt. And if you look at this nerve cell, when this little baby gets stimulated, an electrical impulse is going to proceed down this nerve cell, and as that impulse reaches the end of the nerve cell, these little packets of chemicals, dopamine, are going to move toward the outside membrane and burst. And as they burst, they're going to spray dopamine out into that little gap called a synapse. And if there's enough dopamine out there, it's going to cross over and bind to this membrane, and it's going to fire the next nerve cell in line. And that's how your brain works, neuroelectrical impulses. But the point here is that there's dopamine being squirted outside the nerve cell. Now, do you see this little baby right here? That's a vacuum cleaner. And there are many of them on the outside of this nerve cell, because the job of that little pump, the job of that little pump is to vacuum up all the dopamine once it's done its job. And it's known as the reuptake transporter. And one of the genes I showed you, called DAT1, builds and operates that pump. If you have a longer version of that gene, you have too many pumps on your nerve cell, which means when this nerve cell fires, the dopamine gets vacuumed right back up again. And it can't do its job. And it leaves you in a state of too little dopamine.